Hello everyone and welcome to today's webinar organised by the Pathologist and Ultivue. My name is Rich Whitworth, I'm Content Director of the Pathologist and I'm very pleased to be the moderator of today's online event which will look at leveraging next generation multiplex fluorescent immunohistochemistry. Please allow me to introduce today's fantastic speaker, which is Dr. Alex Klimowicz. Alex is a Senior Principal Scientist at the Department of Immunology and Respiratory Discovery Research at Boehringer Ingelheim Pharmaceuticals. There, he leads the molecular histopathology group in implementing novel in situ techniques, whole slide imaging, and digital image analysis. The group's aim is to build target-to disease linkage in human tissue specimens, a function that underpins projects and external collaborations across the department. In today's webinar, Alex will discuss the possibilities of multiplex fluorescent IHC. He'll explain how Ultivue's InSituPlex platform works, discuss how InSituPlex methods can open doors to new types of IHC-based research, and also share how those methods can overcome existing challenges to streamline discovery research workflows. Alex will be on hand to answer any questions you may have at the end of the presentation, but please do remember that throughout the presentation you can submit questions using the Ask a Question button. And so, without further ado, please let me get today's webinar properly underway by passing you on to our presenter. Alex, over to you.
Thanks, Rich, and hello, everyone. So I'm really pleased today to have the opportunity to discuss with you one of my favorite topics, my favorite research topics, that is, multiplex fluorescence immunohistochemistry. And so, as Rich mentioned, this webinar will start out by exploring some of the challenges that we face in the lab using current multiplexing technologies, and we'll then focus on our user experiences using the Ultivue InSituPlex fluorescence multiplex IHC platform and provide insight and examples of how this technology has been applied in a research setting to address our current challenges, to enhance our research workflow, and to create new opportunities for IHC-based experimentation. So multiplex immunohistochemistry is a powerful tool, and it allows us to visualize the context of target protein expression within human disease tissues. And so unlike other methods, for example, flow cytometry, and not to say anything bad about flow cytometry, it's a fantastic tool, but this provides, flow cytometry is limited and it provides context to your sample type, say healthy versus disease, perhaps your cell type specific context, CD3, CD4 double positive cell. But multiplex IHC adds on another layer, and that's the spatial context within the tissue. So it can provide context of protein expression relative to the pathological features of disease, with respect to other cell types of interest, and with respect to other targets of interest. And the more multiplexing we can apply to immunohistochemistry, the more complex the questions we can ask of these tissues. So here in front of you is an example of some multiplex immunohistochemistry. We're looking at an abscessing crypt in Crohn's disease patient tissue resection. And so that crypt I've outlined here in yellow, and you can see the epithelial cells don't look quite the same as the epithelial cells in the surrounding crypts. There's also some leakage in the epithelial barrier where contents of the lumen of the gut are now able to move into the tissue, and immune cells are moving out into that luminal space. So this is the beginning of a disease lesion. And so now we're interested in our target of interest. Our target of interest is shown in red. And you can see that the context of expression here is very focalized to that pathological feature of disease, that crypt abscess in the epithelial cells and the cells beneath that abscess. But now we have some additional questions. What kind of cells are these? What state are they in? And why are they different than the cells in the surrounding tissue? And so we can now layer on top something like CD3 or CD68 and CD163 in this green channel to look for phagocytes. And we can see that the bulk of those immunoreactive cells are in fact phagocytes that are below this abscessing crypt. And we can see that their morphology is different. So we have this context of shape that we can see. They're spread out compared to the green phagocytes that are in the surrounding tissue further away from that crypt abscess. And so now we can actually probe the cell state. And here we're looking at phosphostat-1 in white. And we can see that the nuclei of many of these phagocytes that are positive for our target of interest are also positive for phosphostat-1, suggesting that these cells are perhaps, in fact, responding to a burst of interferon gamma associated with that loss of crypt integrity, and they're activated in some way. And so in this one image, we've been able to capture a wealth of information to inform our in vitro scientists on how to develop disease-relevant assays to probe our target of interest. So here they could take, say, some bone marrow-derived macrophage or some monocyte-derived macrophage, activate them with interferon gamma, perhaps incubate them in the presence of bacterial contents, simulating this loss of barrier integrity, and then challenge them and use inhibitors of our target of interest to understand the biology of disease in a context that's most appropriate to our disease and some of the features of that disease. Now, the devil is in the details here. For certainly, generating this assay took a lot of time and effort and optimization. And that's something that we run into on a regular basis, using our workhorse in the lab for multiplexing, which is a tyramide-fluor opal-style immunohistochemistry assay. And so what we'll do is, before we go any further, I'd just like to talk to you about some of the challenges that we face with respect to using this workhorse in the lab. It's a fantastic tool, a fantastic method, but it does have some specific challenges that lead to a lot of optimization being needed for multiplexing assays. So to make sure we're all on the same page, I'll just walk you through some simple cartoons of how this method generally works. So here we have our tissue on the bottom, and we're looking for co-expression of two epitopes in the same subcellular compartment. So we throw in our first primary antibody, binds to our epitope number 1. We then go in to detect that antibody with our secondary conjugated to an HRP. And then using the tyramide chemistry and that HRP reaction with hydrogen peroxide, we activate our tyramide-fluor so that it can covalently bind to tyrosine residues in the local environment. So here, we're not looking at the immunoreactivity that we see is not focalized around the specific binding site of our antibody to the epitope, but it's precipitated in the local environment, very similar to a DAB chromogenic reaction. But here we're actually covalently binding our fluor to these tyrosine residues in the local environment. So now, because these are covalently attached to amino acids in the local environment, we can use a short round of heat-induced epitope retrieval to remove our primary antibody and our secondary antibody. Second here. And begin our second round of immunohistochemistry. So here we have our target of interest. We have a secondary antibody with HRP. We use a different tyramide-fluor, and we activate that fluor. It binds and covalently attaches to tyrosine residues in that local environment. Say we're looking for co-expression of CD20 in the membrane compartment of a B cell, and now we're able to see this co-expression, or this immunoreactivity that overlaps. And so this is a really powerful tool. We're able to, because of the ability to wipe away the antibodies, use the best antibody for every target. So if we want to use a bunch of rabbit monoclonals one after another, we can, which is exceptionally powerful. And we're able to amplify the signal for every target. And with a lot of our immunology research, our new targets of interest have very low immunoreactivity levels or very low protein expression levels. And so this is really, really critical for our ability to understand the biology of these targets in human disease tissue. Now, the challenge comes really when we start to look at co-expression of cell type specific protein biomarkers with our targets of interest. And this really relates, I think, a little bit with the chemistry of tyramide and a little bit around the idea of why these are cell type specific protein biomarkers in the first place. And this is often because they have very high levels of protein expression in one cell type versus all other cell types. So we have this very high protein expression, and then they've been around for a while, these cell type biomarkers, and so we have very high affinity reagents for them. And so combined with high protein expression, this leads to some really intense and robust immunoreactivity. And this can be a challenge when you're looking at co-expression in the same subcellular compartment. And I'll just show you a cartoon of why that is. So we have our first primary antibody, our first secondary HRP, and then we lay down our tyramide-fluor. And what we end up seeing is now we've almost saturated all of the local tyrosine residues in this subcellular compartment with, say, our CD20 signal, leaving very little left for our second round of detection. So now that we use our heat-induced epitope retrieval, we wipe away our first set of primary antibodies, and now we're looking at our target of interest, our primary antibody, our secondary antibody, and now you may get the false impression based on this, the fact that there are very few tyrosine residues left to conjugate, that we don't have strong co-expression in this subcellular compartment because there was just not enough tyrosine around to accept that signal. And this isn't the only challenge that we may have with tyramide chemistry. The other issue resides around the epitopes themselves, the subsequent epitopes after our first primary antibody goes down. And so here again, we have our first primary antibody binding, our first secondary antibody, our activation of tyramide. And even if we're not saturating the entirety of all of those local tyrosine residues, what can happen is you can covalently modify one of the tyrosine residues that may occur in one of your subsequent epitopes. And so this covalent modification impacts the ability of your second primary antibody to either recognize that epitope completely or reduces the affinity of that antibody for that epitope. And so again, you get this false impression that there's weak to no signal in that cell type specific compartment. And so you may wonder, well, how often are tyrosine residues found in IHC antibody epitopes? And the answer is more commonly than you think. So this really nice paper by Sampurin et al. In 2006 evaluated the epitopes of a number of the different clinical IHC antibodies that were being used at the time. So these involve many of the ER, or estrogen receptor, progesterone receptor, HER2 clones, the MIB1Ki67, and the DO7P53 clone. And when they looked at the frequency of different amino acids occurring in these epitopes, what they found was that the third most common amino acid was a tie between glutamine and tyrosine. So this does happen, and we do see this happen, and I'll show you some examples where this may be happening. So while there are some challenges with tyramide chemistry, that's not to say that they're not insurmountable. So I'll just give you an example. So here we're looking at an assay where we're looking to co-express our target of interest with CD20 to look for B-cell co-expression. And I'll just give you a warning. Right now, most of the fluorescence images I'm going to show from now on in the presentation have an inverted background. And so I like to invert the background from black to white. I feel I can see a lot more, and it's much easier to present with this white background. So this is not bright field immunohistochemistry. So what you're seeing now in red is our target of interest, and this was applied after we did an anti-CD20. In green, I'm not showing the anti-CD20, I'm showing the impact of anti-CD20. And so you can see in the left panel that if we were to take this at face value, we would say that our target of interest in red is expressed in the inflamed mucosa of an ulcerative colitis patient, and perhaps in some of the muscle around blood vessels, but certainly not in the B-cell areas of gut-associated lymphoid tissue, sort of central in that image. But if you then run the appropriate control, which is running an isotype instead of your anti-CD20 antibody, followed by our target of interest with a tyramide reaction, you can see that, in fact, the most robust immunoreactivity that we see is in the B-cell areas of gut-associated lymphoid tissue for our target of interest. This discrepancy was caused by interference with the tyramide chemistry. And so we were able to optimize this out, and it seems like a very simple solution. It took us a while to get to this, but you can see on the left panel we have the impact on our target of interest in red with the 1-to-1 dilution of the ready-to-use antibody we were using for CD20. And then once we diluted that out to 1-to-15, you can see the impact that it has on immunoreactivity of our target of interest in the B-cell area of gut-associated lymphoid tissue. So it's really night and day here. So by diluting this down and perhaps either not modifying tyrosines in the epitope or by not saturating all of the tyrosine residues in that membrane compartment of the B cells, we're able to see some really nice, robust immunoreactivity. And we were still able to co-localize this with CD20 in green. Here we benefit from the fact that we can use a longer exposure time, even though the signal is weaker with that 1-to-15 dilution. And it turns out that our CD20 antibody was such a good antibody that we did have a lot of signal to noise, even though we had this reduced concentration. This doesn't always turn out to be the case, and this is sort of where we stand with some of the challenges of that. Sometimes you just don't have that signal to noise, and you start to lose your cell type specific immunoreactivity at the expense of trying to rescue the real immunoreactivity that you should be seeing for your target of interest. So just to briefly summarize some of the challenges we have developing co-expression assays with Tyramide, while it is an awesome assay, it really creates a lot of flexibility in your assay design. It also involves a substantial investment in assay optimization time for any given combination of antibodies. And I've just listed a few things here that we consider. So the order of primary antibody application, which fluor should we use for which target, the concentration of our primary, as I showed you, the concentration of the Tyramide fluor itself. And then one of the most obnoxious things that we encounter is sometimes you've developed this beautiful 3-plex, 4-color assay, and you want to swap out one of the targets because you want to really address a different but very specific question, and you end up having to re-optimize the entire assay because it just doesn't fit with that optimization that you generated with the original antibody. We also see some limitations to this system that would be nice to be improved if we could going forward with next generation platforms. And so that is that the ability of our fluorescent filters to separate target-associated fluors. We're depositing covalently these targets on here, so we can't remove them. And we need our filters, because we use a very standard fluorescence microscopy setup with a number of different instruments that are all filter-based to separate our signal. And right now, our limits are really, we can separate 4 channels without any bleed through. And we've tried a number of ways to get around this, but we're really stuck with those 4 channels to be able to sensitively detect the signal and to not have bleed through from one channel to the next. In addition, and something that we don't normally encounter, is that sequential heat-induced epitope retrieval effects can impact the tissue histology. And so as you get up past 60 minutes of total heat-induced epitope retrieval, as you're wiping off your antibodies, you can start to impact how the visualization of the histology of the tissue. And there's certainly methods to address these limitations, but these require time and/or specialized equipment or expensive equipment. So, things like multispectral imaging, which can maybe bring you up to 8 or 9 channels and cleanly separate those channels, or multiplex ion beam imaging. But right now, we were really focused on finding methods that would use the equipment and the infrastructure that we built up using sort of standard fluorescence microscopy whole slide imaging techniques. And this is where we came across at a conference, the Ultivue platform, the Ultivue InSituPlex platform. And so we were at a Leica symposium when I first saw this technology, and they'd optimized this using the BondRx, and this just happens to be the instruments that we use. And so we're really excited to see how they were taking immunohistochemistry and transforming that method into something that was easier, more flexible, and more powerful, that addressed our very specific challenges around this co-expression IHC. And so here, the Ultivue InSituPlex platform replaces secondary antibody detection with a DNA barcode on your primary antibody. So here we have 4 different antibodies recognizing 4 different targets, and each one of them would have a distinct DNA barcode. And so because they all have distinct DNA barcodes, we can stain with them all simultaneously. So we're putting them on all in one step. Then we amplify those barcodes. It's all just amplification of DNA. So this again happens in one step. Once you've amplified that DNA barcode, you can add a mixture of fluorescent probes. Each probe has a different fluor, and each fluor is then targeted to a different barcode. So we can now visualize 4 targets simultaneously. So what you can see already is that this has greatly simplified the workflow for multiplex immunohistochemistry. We're not cycling back around doing one set of antibodies, heat-induced epitope retrieval, and then a second set. We're actually able to stain everything simultaneously at simple steps. So it makes setup a whole lot easier, and it reduces the amount of time. Our sort of homebrew opal-style Tyramide method for a 3-target, 4-channel assay was taking us a full day and then an overnight reaction, and then we come in and finish it in the morning to visualize on our whole slide imaging devices. This assay now takes somewhere between 5.5 hours, and reduces the amount of error that you can introduce because it's a very simplified setup. And I think most importantly for me is that it has the potential here, when we saw it, to avoid the challenges associated with tyramide chemistry. So it enables effective co-expression immunohistochemistry using standard methods in slide scanners, because now we're not using tyramide. The only downside to this is that if you want to look at your own specific targets, you do require an Ultivue DNA barcoded antibody. It's easily addressed talking to the company. They're very willing to custom label antibodies for you and custom develop kits. Although currently there are 4 InSituPlex kits that have been released and are commercially available with predefined antibody combinations, and they've been validated and optimized for use on BondRx autostainers as well. So all of these are focused around the immuno-oncology space. So there's the PD-L1 kit, a PD-1 kit, an antigen-presenting cell kit, and a T-cell activation kit. And so over the next set of slides, I'm going to walk you through some of our experiences with alpha and beta testing each of these kits, and some of the experience that we have and some of the insight that we have from this experience. All right, just a sec here while my slide changes over. So just to summarize, the method itself, the InSituPlex assay streamlines our IHC workflow on the BondRx from an overnight assay, all day and an overnight, to something around 5 hours. The method also allows us, I didn't really focus on this, to use the size 7 channel or something further into the infrared. I call it the size 7 channel, but I'm not sure which fluor is actually being used here. This is not easily or typically detectable using HRP and a tyramide size 7, and trust me, we have tried this. And so this was really an extra bonus to this assay system, is that we're now increasing our level of plexing using the same traditional filter-based fluorescence microscopy techniques. And then the assay kits were very well optimized and easy to use. And so over the next few slides, I'm going to walk through our experiences and some of the insight that we have around the fact that the signal that we see provides a higher resolution of expression of our targets compared to HRP-based amplification. Around the co-expression of multiple targets in the same compartment, they don't appear to suffer from the same challenges that we've seen in the past with tyramide-based multiplexing, and how we can simplify our assay workflow from a discovery mode where we look at a large number of targets simultaneously, identify something that we're very interested in, and then switch over to a smaller, more focused production mode to analyze a number of samples with that very specific set of antibodies. All right, my slides are just taking a few seconds to load here. So one of the first things that I noticed when we started using the Ultivue platform, and this is using the T-cell activation kit as an example, was the nature of the immunoreactivity. So traditionally, when you're doing Ki-67 with a DAB reaction or with a tyramide-fluor reaction, you expect to see a diffusely nuclear signal, just like you do on this tissue here. This is, again, a piece of Crohn's disease patient tissue in the gut. And so you can see this nice brown diffused nuclear staining. Sometimes you do see a few spots here and there. I always thought these were more about nucleoli and subnuclear structures. But what you see with the Ultivue platform is more of a punctate spot. And while I cannot claim, and I'm not sure that each spot represents an individual antibody, certainly what we see is a more focused immunoreactivity around the site of our antibody binding. And this threw me off at first, when we're looking at the Ki-67, because I wasn't used to seeing it as these punctate spots. But we go from, sort of a weaker Ki-67, which is more of a dispersed set of spots, to a higher concentration of spots in a local area, higher density, as a more intense staining in some of these nuclei. And this makes sense when we think about the platform itself and the technology behind it. So as opposed to precipitating or activating a fluor and depositing it in the local area, here the DNA barcodes and the amplification of those barcodes holds the signal proximal to the area of primary antibody binding. And while you may look at this cartoon for the amplified DNA barcode and say, well, that's pretty big and bulky. In fact, speaking to the Ultivue scientists, they tell me that the amplified barcode is usually smaller than the Fc region of the antibody itself. So it does provide a lot of amplification in that very small space, but it's very tightly localized to where that primary antibody is binding. So we get this much more high-resolution signal. And so right now we're exploring a number of different applications in the lab where we might be able to take advantage of this high-resolution immunoreactivity to provide subcellular data, subcellular localization data. And then I think most importantly for me, from thinking about assay development and optimization, is the idea that the Ultivue system avoids the tyramide chemistry associated problems we've had around co-expression of targets in the same subcellular compartment. So here I'm showing you the PD-1 kit. It's in gut-associated lymphoid tissue in a patient with Crohn's disease. And so there's no pancytokeratin in purple. We're really just looking at the T-cell markers here. And we're starting out with PD-1. And you can see that the PD-1 immunoreactivity has a broad range of levels from very, very faint to much more intense. If we next overlay on top of this our CD45RO signal, we can see that it has the same type of dynamic range from very faint to very intense, and you can match this with varying degrees of PD-1 immunoreactivity, and there does not appear to be any bias that when we have stronger blue CD45RO, we see weaker red PD-1, which was a good step in the right direction. And I think perhaps most impressively is when we layer on top of this our CD3 immunoreactivity, which is just really robust. We still are able to see some cells have very weak PD-1 and weak CD45RO all the way through robust. So the fact that we have 3 targets that all can localize in the same subcellular compartment of the same cells, and we're not seeing any of these biases or bleaching effects like we saw with our target of interest with that CD20 assay was really impressive to me. And so I'll just zoom in a little bit, give you a higher magnification view. This is at a site of mucosal barrier injury in another patient with Crohn's disease. And again, you can see that there's really nice dynamic range from very faint immunoreactivity to very robust. And again, you can start now with the higher magnification to see that more punctate nature of the immunoreactivity. And that higher intensity is really a higher density of these punctate spots. And so we layer on top of this CD45RO, and again, you can see this really nice dynamic range. And then we layer on top the really robust CD3. And again, there does not seem to be any difference between, or any bias in the CD3 causing artifacts in the CD45RO or PD-1 immunoreactivity levels. We see a whole dynamic range from very weak in some of the center cells to very robust PD-1 and CD45RO in the top left corner. I'll just walk you through a couple of the other kits. This is the antigen-presenting cell kit, looking at CD20 in pink, CD11C in blue, CD68 and CD163 in the red channel, phagocytes, and MHC class II. And so again, we're looking at gut-associated lymphoid tissue. And you can see that our phagocytes are distinct from our B cells. And then we layer on top CD11C. We can pick up some beautiful co-expression. This looks black when we overlay the dark blue and the red. So there's a subpopulation of phagocytes that are CD11C positive within this gut-associated lymphoid tissue. And then when we layer on top MHC class II, we can see that there are some cells that are just class II positive, and negative for the other ones. Some are triple positive, phagocyte CD11C, and we even see some co-expression with CD20 and MHC class II as well at a fainter level or a weaker level. And then this picture here is from a patient with idiopathic pulmonary fibrosis. And we're looking at alveolar macrophage. And this was a particularly striking image to me because it really shows the biological diversity of these alveolar macrophage in the lumen of the lung. All of these cells in the middle of the tissue and on the far right are alveolar macrophage. And you can see there's this beautiful array and gradient of color from just plain yellow with MHC class II all the way to really robustly red over top of that MHC class II to really robustly blue over top of that, or some combination in between. So while this is really nice, we've got these 4-plex assays, it really empowers us to ask more complex questions in our tissue. We're still left with a lot of assay development to look at our targets of interest within a broad array of different cell types. And a lot of our new targets of interest, there's very little biology out there, there's very little published out there about where they're expressed. And so we really want to cast a broader net on different cell types. And so as a proof of concept, we've worked with Ultivue with something that's not quite, not commercially available yet. They call this the exchange reaction. And they've published this in a couple of really nice posters at some recent meetings. And so this really involves combining multiple 4-plex reactions together to create higher-plex assays. And so here we worked with Ultivue to develop as a proof of concept that we could perform this in our lab, the PD-1 kit combined with the PD-L1 kit. And so here we have 8 antibodies. Now, technically, this is an 8-plex reaction. There are 8 antibodies, but in fact, both kits look at pancytic keratin and one antibody. They have the same antibody doing this. So in fact, we get a 7-target, 8-color assay in the end. But regardless, we put all 8 antibodies on. Each one of them has a unique barcode, so we can stain them simultaneously. We can amplify all of these barcodes simultaneously. And then, because we're using a standard fluorescence microscopy setup, we can't visualize them all simultaneously, so we visualize them 4 at a time. And so here we take the first set of probes that recognize, say, the PD-1 kit, we image them, and then we pop off the cover slip and then use a gentle enzymatic process. It takes about 25 minutes to remove the first set of fluors, and then rinse that off and add our second set of barcode-specific fluors for the second kit, say the PDL1 kit. Once we've imaged that, we then take the 2 sets of images, we stack them together and align them using the Herbst or DAPI signal, and now we've created this 7-target, 8-color fluorescence immunohistochemistry image. So a very, very simple idea and a really powerful tool. And so I'll just give you an example of what this looks like in some human tonsil tissue. So in this tonsil tissue, we'll start off with pancytic keratin. So this is where there were actually 2 antibodies, 1 in each kit, and I've layered them together. And so you can see this really nice yellow signal for the epithelium in the tonsil. Next up, we'll look at CD68 to detect phagocytes within the tonsil. You can see in dark blue. And both of these cell types can co-express PD-L1. You can see there's some really robust PD-L1 in the epithelium up in the top left, and a much more moderate PD-L1 in the phagocytes, particularly in the germinal center. And you can see it's a very membrane-localized signal. Now, these are not the only cells in here, so we're also able to look at CD3 cells in cyan. And some of these CD3 cells are co-expressing CD8 in brown. Not very many of them. And then we can layer on top of that CD45RO, suggesting that most of these cells are more of a memory phenotype. Most, but not all. There's the odd one. There's one down at the bottom left hand of the screen that doesn't have any CD45RO. And then finally, we layer on top of this PD-1. And so now we have an ability to address much more complex questions and interrogate a much broader diversity of cell types for our targets of interest and their expression patterns within human disease tissues. Now, this does take more kits. It does take a little bit more time, and certainly there's much more information to analyze. So if we want to move from a discovery spot with an exploratory project that we're moving through our project pipeline, and now we're at a stage where we want to evaluate a very specific question that we've been able to pick out using one of these discovery assays, we now want to be able to sort of streamline the assay down to at least a single 4-target assay or maybe even less. And so one of the scientists in my lab decided that this would be a great thing to try. And one of the questions to ask would be, how do PD-L1 and PD-1 expression relate to each other in different inflammatory states? And so what she did is she actually took the PD-1 kit, the PD-L1 kit, and because the commercial kits are initially produced with the same barcodes, she just swapped out the CD3 antibody from the PD-1 kit and replaced it with the PD-L1 antibody from the PD-L1 kit. So after that straight and simple swap, she ran the standard assay, and we were able to generate, without any assay optimization, a very comparable result to that 8-plex assay, but really now focused on 2 targets of interest, PD-1 and PD-L1, that were in separate kits to begin with. So this is really a proof of concept that once you've optimized the assay for Ultivue barcoding and it's been put into one of these assay kits, you should be able to just take those antibodies and create custom kits, mixing them together. And this is something that we're pursuing with Ultivue right now. So you can see, again, the signals look very similar to what we saw with the exchange reaction, 7-target, 8-color. Now we're down to 5-color, 4-target, but very similar immunoreactivity patterns, very similar quality. now we've sort of generated this proof of concept that we can move a 7-plex assay down to a streamlined 4-plex assay. So now we wanted to sort of run through our the exercise of analyzing some of the questions that we could ask from this tissue. And so this really related, we thought it would be interesting to look at the relationship between PD-1 and PD-L1 immunoactivity, given that they're a nice receptor-ligand pairing. And so the analysis that we did was using the Halo image analysis software from Indica Labs. And so here we're looking at PD-1 in green. And we used the area quantitation algorithm to set a threshold and create a PD-1 mask. We did the same thing for PD-L1, created a PD-L1 mask. And so then when you look at the two signals together, PD-1 and PD-L1, and while you can see that there is some yellow overlap between red and green in this black background, it's not as easy to see as when you compare the overlap between the two masks that we've created. And so from this, we calculated the percent overlap, the sort of co-expression in yellow, as a percentage of the total PD-1 area to try and gain an understanding of what percentage of PD-1 is engaged or potentially engaged with PD-L1 at any given point in different states of inflammation. And this is a contrived assay. Certainly, we don't have the resolution to be able to say that there's an actual interaction between PD-1 and PD-L1, but we consider this to be a conservative overestimate of the amount of PD-1 and PD-L1 interacting, which was sufficient to ask our question. And so here, when we ran this on a number of different germinal centers in human tonsil and analyzed them, what we found was that on average, 19% of the PD-1 positive area was also positive for PD-L1, suggesting that somewhere less than, or somewhere greater than 80% of the PD-1 is available for interaction at any given point, and that there's the potential for somewhere less than 20% to be interacting with PD-L1. So, in summary, I think the InSituPlex technology is a fantastic improvement upon our current level of multiplex IHC, or current technology for multiplex IHC, and enhances our workflow. It reduces the time needed to run these assays. It reduces the complexity of running these multiplex IHC assays, and it provides us an additional target that we weren't able to use using a standard fluorescence microscopy setup. In addition, this also, the signal that we develop provides higher resolution immunoactivity. That signal seems to be limited to the area immediately associated with our primary antibody, and this may open up some avenues and some applications where we can now evaluate with more confidence subcellular localization of specific targets. It also appears to address our challenges that we've had with Tyramide co-expression assays, streamlining the development of these co-expression assays. As you saw, we were able to just sort of pop antibody from one kit to another, and without any optimization, look at the overlap between them. It also enables more flexibility to obtain flow cytometry-like cell expression analysis in tissue. We can layer on top at least 3 different targets or 4 different targets in the same subcellular compartment, and we don't seem to have any kind of interference between them. It also streamlines our workflow from this higher-plex discovery assay system into a lower-plex production assay. As I mentioned, the exchange reaction enables stacking of these 4-plex assays, and it works exceptionally well. We were really impressed with the combination of the PD-1 and the PD-L1 kits together in the same image. And then we were able to streamline this down to a production higher throughput assay with a single 4-plex by mixing and matching antibodies to create these custom assays. So overall, we're very happy with the next generation technology for immunohistochemistry that the InSituPlex from Ultivue provides. And just a quick disclaimer, the opinions expressed in this presentation are solely mine as an expert in the field and should not be considered an official endorsement by Boehringer Ingelheim of any product or service described herein. And so with that, I'd like to thank my team at Boehringer Ingelheim, in particular Marina Bleck and Diane Mears, the scientists who created all of these beautiful images and ran all of this image analysis and immunohistochemistry. and our director and one of the project leaders, Jerry Nabozny. I'd also like to thank the team at Ultivue, Louis Levy, the head of business development, as well as a fantastic group of scientists that we've had the pleasure of working with, Amanda Bars, Katir Patel, Bonnie Phillips, Jamie Buell, and Courtney Hebert. And with that, I'd be happy to take any questions. Thanks very much for your attention.
Thank you so much, Alex. This is Rich again. You showed there what is clearly a highly innovative multiplex IHC platform out in the wild. As you noted, we've received a number of questions from the audience. Please do send your questions in, because any that we don't have chance to answer during the live webinar, I'm sure the team or Alex himself will get back to you with some answers. So Alex, are you ready for the first question from the audience?
Absolutely.
This is a simple one, I think, although I guess it will depend on the particular assay. But they say, how much time does it take to optimize a multiplex IHC assay using the Ultivue technology?
Well, thanks, Rich, and that's a great question. I think, as you saw in the presentation, we were able to take 2 4-plex kits and swap out antibodies because they had compatible barcodes, and so that took no optimization time. That being said, I think that would rely on you having kits available with targets of your interest. So it might take you a little bit longer to optimize a fresh assay, because you would have to optimize your IHC conditions for that single antibody and send it to Ultivue for conversion or labeling and enablement with their assay system. That being said, it seems like once you have a repertoire of antibodies, we should be able to, in theory, mix and match them very quickly to develop assays without much in the way of optimization time.
Great, thank you, Alex. Here's a slightly more specific question. They say, would this technology allow us to identify dim versus bright expression of T cell activation?
Absolutely. So you saw in a couple of the images there was PD-1 levels were very low or very high, and the CD45RO also had this beautiful dynamic range in T cell compartment. And using the halo image analysis, we were able to separate those two up. I didn't present that today, but that's something that is quite feasible.
Here's another question. Something I think you didn't delve into. They say some questions might require interrogation of RNA targets with protein targets in a multiplex panel. They say, do you have plans to do this, or can you offer any speculation as to how well this might or might not work?
I absolutely agree with that question. That is certainly something that's part of our normal workflow and what we've been using with the Tyramide chemistry. I'm not sure. We have not tried that yet, but that is certainly on our list of things to do. And I'm quite hopeful, just even thinking about the technology and the compatibility between the two, say an ACD RNAscope assay followed by an Ultivue assay or vice versa, that this in theory should work well, but I would have to defer to the scientists at Ultivue because they may have some more information around this. But this is certainly something that we're planning on trying in the not too distant future.
Great, thank you, Alex. I'm sure the Ultivue team can get in touch with that individual. Here's a question, actually really looking at the tissue. They say, what type of preservative do you recommend for human samples, I guess, when doing this kind of multiplex assay, i.e. heparin, EDTA, formalin, etc.?
So all of this is, I think, based on formalin-fixed paraffin-embedded tissue. So the antibody clones are optimized for use. These are just standard IHC antibody clones that the Ultivue team has been able to label and optimize the assay on FFPE tissue. So it may work beyond FFPE tissue, but right now my understanding is the kits are for FFPE tissue. All of the tissue that we've tested is formalin-fixed paraffin-embedded.
Thank you. Here's a sort of question comparing technologies, and I guess this will be somewhat dependent on whether or not you've used the technology they've chosen. But they say, how does this Ultivue InSituPlex fluorescence platform compare to the imaging mass cytometry on the Hyperion imaging system that can interrogate up to 37 different protein markers simultaneously? I guess my answer to that would be, I guess the second system is considerably expensive, but probably you've got a little bit more insight, Alex.
No, that's really the insight that I have. We haven't tried that out, that Hyperion technology out, or the multiplex ion beam imaging, a very similar technology. right now that's beyond the cost that we can bear. So the really nice thing about the InSituPlex system is that you just buy a kit and it works with your existing infrastructure. So it's a really good question. Obviously, you're not able to multiplex 37 targets simultaneously yet with Ultivue, but I think it's a big advance compared to what we have been using.
And I guess with 37, that's a lot of data crunching in any case.
Yeah.
But
yeah, you probably have a whole new set of challenges at that point.
Absolutely. Okay, let's move on to another question. This is actually quizzing the technique, so once again, stand by for this question. Are you sure that the barcode-specific complementary DNA probes are specific to the amplified barcode and do not recognize any DNA from the sample? A good question, but I feel sure that Ultivue would have thought of this.
Yes, and I will have to defer this question to Ultivue. From what we've seen, we've not picked up any background with the images that we've run. We've also run some negative controls where we, instead of using the barcoded antibodies, we use unbarcoded antibodies on the same pieces of tissue, or serial sections, and then we run the whole assay with an amplification reaction and with the barcode-specific fluors, and we pick up absolutely no signal. So I would suggest from that yes, they are absolutely specific to the amplified barcodes. But again, I can defer you to Ultivue for more details.
Great. Thanks, Alex. Here's a good question, I think. Have you had any chance to do any 3D evaluations of Ultivue multiplex samples, for example, with serial sections or WF 3D imaging?
And not at this point, no. But that does sound like an interesting approach.
Question here they say, is it biotinylated tyramide signal amplification, and how much BTR or tyramide signal may or may not interfere with other antibodies? I guess here they're going back to maybe your original discussion.
Yes. So our workhorse in the lab is not using the biotinylated tyramide, but just the tyramide fluors. That extra level of amplification hasn't been necessary for our applications. So I can't really comment on biotinylated tyramide signal amplification.
Great, thank you. This sounds like a good question. Why wouldn't you use fluor conjugated primary antibodies or fluor conjugated secondary antibodies to avoid some of the challenges associated with tyramide-fluor multiplexing?
That's another great question. And we have in the past done this, but the challenge here is a lot of our targets are very weakly expressed. And so we don't get enough signal to noise. We'll pick up background autofluorescence in the tissue when we use these more directly conjugated methods. And so that's where the Ultivue method, it gives us that same sort of signal resolution, but a much, much more robust signal.
Thank you, Alex. Here, once again, this will very much depend on what you've experimented with, Alex, but they say how much variation is there between fresh frozen or paraffin embedding section and rat versus human tissues if you're using Ultivue's technique?
So I don't have any experience with fresh frozen yet. Again, all of the antibodies are FFPE optimized antibodies. So all of my experiences with paraffin embedded sections. And then we have not looked at any other species other than human because the existing kits are all focused on human-specific antibodies. And we haven't tested whether or not they're cross-reactive with rat, mouse, or other species at this point.
Great, thank you. Someone here missed a point. They say, is it only fluorescence or is it also chromogenic?
So this is a fluorescence technology. The probes are labeled with fluors. The barcode-specific probes are labeled with fluors. Great,
thank you. This is a challenging question to answer. And I know you said, I think at the beginning, research use only, but they say, could this method, and I guess this is a sort of, in the future, could this method be made economically feasible for clinical application?
I think, hypothetically speaking, I think the answer there is yes. I think this is another good question to bring up with Ultivue. Yeah,
Great, thank you. And someone asks, can this method detect intranucleus proteins?
And I think the answer here is also yes. We've only really had experience with the antibodies that have been produced in the existing kits. And there we see those punctate spots with Ki67, which is a nuclear protein. So this is certainly something that we're exploring. How can we look at that intracellular subcompartment analysis with respect to IHC using the kit?
Great, thank you. This is a good question. They're obviously paying attention. If the primary antibodies can be swapped out, PD1 combined with PDL1 kits, how do you get specific fluor binding when you combine the two kits in one assay for the 7-target, 8-color assay?
So to do this, so the commercial kits were initially produced with the similar barcodes. When we worked with Ultivue for that exchange reaction, they had to relabel one of the kits with unique barcodes. And so, again, I'll have to defer to Ultivue on how this is going forward, but I think my impression is that they will start putting unique barcodes on all of the antibodies. Great,
thank you. This is someone wanting more comparison, Alex, so you may need to invest in some more kits so you can do back-to-back comparisons. They say, did you make a back-to-back comparison with the Opal technology?
So we haven't actually done that. That's a really good point. I know Ultivue, during their method development, does comparisons, DAB to fluorescence to Ultivue InSituPlex. We did not make those back-to-back comparisons because we don't use a lot of these antibodies and those specific clones. And so we would have had to invest in a new set of clones. For us, the advantages that were provided by the InSituPlex were sufficient for us to say this is a new system, a great new system going forward that we should really try and adopt because it addresses a lot of the challenges that we currently have.
Well, thank you very much, Alex. I think that's all we've got time for now. There are a few questions left in the inbox. So as I said before, either Alex or the Ultivue team will be in touch with those people. I should add that this webinar, the whole webinar, including the Q&A, will be available on demand. So if you want to rewatch a portion or want to go back over Alex's rather wonderful slides, then you'll be able to do that. We'll send a link out to the whole audience once it's available. Likewise, if you have colleagues or someone else that could benefit from this webinar, or you think would benefit from this technology, please do share that link on, and we can spread the word a little bit wider. I'd just like to thank you once again, Alex, for your time and insight. It was really great talking to you, and I'd like to thank everyone else for joining us today, and it just leaves me to say I look forward to seeing you all at future webinars. Thank you and goodbye.